Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 204 for the podcast. Today, I'm sitting down with Randy Teel, President and CEO of Arvinas, a clinical stage biotech based in New Haven, Connecticut, that's doing something genuinely new in medicine. Arvinas is pioneering a technology called PROTAC protein degradation. They've got a platform that doesn't just block disease-causing proteins, but eliminates them entirely, using the body's own disposal system to do it. It's a fundamental shift in how we think about drug design, and it's now reached a historic milestone, the first PROTAC ever to succeed in a Phase 3 clinical trial, with a regulatory filing now in front of the FDA. Randy was appointed CEO earlier this year, bringing over 20 years of biopharma experience and a deep strategic understanding to the company he's helped to develop since 2018. This is a conversation about bold science, smart leadership, and what comes next. Let's get into it. Randy, welcome to Beyond Biotech.
Thank you, Dylan. It's great to be here this morning.
You've been with Arvinas since 2018. You were there for the IPO. You led the strategy, the business development. Now you're the CEO. Before we get too much into the business and science of what you're doing there, maybe you can just start by introducing yourself. Tell me a little bit about how you got yourself to where you are.
Well, it's a long and winding road, Dylan, and I think anybody who tells you that they had it all planned out at the start is probably fibbing a bit. So if I go way back, I started my career in the lab actually as a graduate student in immunology. Did that, got a Ph.D, and then very rapidly realized that my place of excellence was less so in the lab, and looked for opportunities, to be quite clear, that didn't require me to do a postdoc. And about that time, I got interested in management consulting, largely because they would come to recruit at the universities. And so I joined a management consultancy for 7 years before jumping sort of back to the science side, back to biotech. And I think that dual history, sort of both as a PhD scientist, as well as squarely on the business side working for McKinsey, I think gives me a pretty balanced view of how to look at biotech. And I think that's carried me pretty well through my subsequent endeavors. I went to Alexion first, was there for 3 years, ended up as head of strategy, and then moved over to Arvinas back in 2018. And as you pointed out, at that point, it was a private company. It was a preclinical company. It's a platform company, something I'm sure we'll get into over the course of this discussion. And started off at Arvinas in a role doing some things I'd done before, like corporate strategy, and some things I hadn't done before, like investor relations and being close to the investor side of biotech. And that's actually why I had come over to Arvinas in the first place, was to get closer to the sort of nuts and bolts of how the industry worked at the earliest level, that intersection between science and business. And so since I joined Arvinas, I've done business development and IR and comms and strategy, commercial for a bit. I was the interim CFO for a bit. And then became CEO earlier this year. So it was a winding road, but it's really been educational along the way, seeing a lot of different sides of the business.
I'm interested in that transition to CEO. You said very recently, it was back in February of 2026, you came into the top role there and replacing a leader who's already there in a company. That's never simple. How did you approach that transition? What did it feel like on day one?
The transition was, we had an opportunity to announce the transition somewhat early, right? So John Houston, who had been the CEO since late 2017, with a phenomenal background at Big Pharma and building discovery groups, did an amazing job sort of building the company from the platform that we had to the pipeline that we had. And then last summer, after the positive pivotal data for our lead program, we announced that John would be stepping away and transitioning after we found a new CEO. And so at that point last summer, we really telegraphed to the world that we would be going through some major transitions. We had decided at that point we'd be transitioning CEOs, we'd be transitioning strategy from really being an almost commercial company focused on commercializing Vepdegestrant to focusing on our pipeline. And that made for a natural transition point. I think that I was told many times that nothing really prepares you for the CEO role. Like at least 4 months in, I can confirm that is the case. So I think, that first morning, in terms of day one, you really feel the responsibility, right? Look, I was the CBO before, so I was not a stranger to important decisions that affect the direction of the company. I played a key role in that decision to not commercialize Vepdegestrant when it was ultimately approved and to shift our focus to the early stage pipeline. But I think as CEO and overseeing the whole company, I would say the weight of those decisions feels very different. I think the every decision that a CEO makes is a hard decision. If it was easy, it wouldn't get to me, right? And so I think, and I just told the company this yesterday, the definition of a hard decision might be one that makes very few people happy, no matter what you do. And I think that's true in every case. We're going to talk a bit about programs. We're going to talk about capital allocation. Allocation means moving funds and resources from one area to another, which means that someone's getting more and someone's getting less. And I think the weight of how that impacts the teams and how they feel and how bought in they are to sort of move forward with the whole company in mind, you feel that weight as the CEO. And it's really important to me that the whole company sort of understands the decisions that we're making. And that is a concern that affects everybody but the CEO a bit differently. So.
You mentioned you did your PhD, you were in the lab, and then you jumped over into this business consulting, and you've been working on the business and strategy side since. Now you're at a company which is very much science driven. How do you actually earn the trust of the scientific teams when you're coming from, the business side, the capital side?
Yeah, I tell our scientists that yes, I have a PhD, but it's a bit dusty. It's been a little while. It's been over 20 years now. So what I think that lets me do is it does give me some credibility. I think that's true. And by the way, that was true in the consulting world too. When I would do work with biotech or pharma or other healthcare companies, it helped that I had a deep understanding of the science. And I think it does help in communications too. Being able to communicate with folks on the business side with the scientific understanding is very helpful. And honestly, what I do is I ask a lot of questions still. And I try to convey and make it very clear that I don't know everything, although maybe that's easy to convey. I ask a lot of questions. We even just yesterday had a internal R&D day with the company with scientists from all sorts of different groups presenting their work and sharing it with the company. And I ask a lot of questions. And I'm very curious and I want to know how things work. And I want to know answers at a detailed level. And I think that means that even when I zoom back up again, to the 60,000-foot level, people know that I've taken an effort to understand the details at the right time and with the right level of intensity. And I think that does help. And it's not really because I'm trying to show anything. It's because I really am interested. I mean, it wasn't an accident that I ended up in a PhD program. I love the science. Like I said, when I transitioned from Alexion to Arvinas, part of that was my role at Alexion was amazing, loved it. But it was very much on the business commercial side, pricing, life cycle management. Very good problems to have, but I was interested in the problems around, well, how does a drug actually get made? How does it move through the stages, getting from discovery to development? What are the pitfalls along the way? How does that work? And at Alexion, that wasn't my role. That's just not how corporate strategy engaged, at least on my side. And here it is, but that was a very intentional choice to come to a place where science is really leading the way.
You came in from that CBO role, so you weren't a stranger to the company. You weren't being parachuted in from somewhere else. But were there things as a CBO that you couldn't fully act on that now become priorities as a CEO?
Sort of. I think that it's really easy in a non-CEO role to look at the CEO and say, oh, they should do this, they should do that. The decision is simple. It's clear. It's clear. The decisions that a CEO makes are never simple, as again, like I said before, but if they get there, they're by definition hard. And so there were things with my general bias towards action that I wish we could do faster. But I'd also say that we are now doing exactly what we said we would do, right? In the wake of having some initial targets for our technology in breast cancer and prostate cancer, we had lit off a number of phase 1 trials that we wanted to see the data for. We said those data would be coming in 2026, and now it's 2026. So there might have been things that I would have loved to make a decision on last year and been antsy to do. But in reality, you have to let the programs reveal themselves and show data because as much as we're driven by what we show preclinically, you have to be to put programs into the clinic. You then have to prove it in the clinic. And you have to make sure that in the phase 1 trial that you run, you get to the answer. And we've always known that that was going to come for a number of programs in 2026, and it is, and it will. And so I think there was probably a bias towards action that I would have loved to exhibit, that now is the time. Like now is the time in 2026 and 2027 to really decide where Arvinas will be the ones to invest in the programs that we have versus where we might look for help from a partner.
You know, I was talking to a CEO for another episode the other day, and he said, you know, there's every time you get someone moving into that chair, they inherit momentum and they also inherit some challenges there. Maybe you can give me an example of each. What's something you're really excited to be accelerating, and what's something that was just keeping you up at night?
Yeah, look, the programs and the people, right? So the momentum is, as I just said, we've come into this year with a number of trials ongoing where we know we're heading towards data, where we know we're going to make big decisions. Like, to me, that's the job. Like, that's a great problem to know we're going to make difficult choices based on where we're going to invest. That's what we've set ourselves up to do. And those will be hard, but I'm excited to do that. I think the challenge is what I said before about that feeling on day one, right? Where you know the decisions that you're making, there will be haves and have-nots. There will be places that we choose to go versus where we choose to get help. And it's really important to me that we move as a company in that direction and people are bought in, because I think, great people in biotech can work anywhere, right? There's a lot of companies out there. Even if you're in Connecticut, we're competing for the best talent in the world. And that's one reason that even post the pandemic, we've kept a hybrid remote sort of structure so that we can make sure we get the best people. But it's really important to me that people are excited to be here. Like Arvinas has been, we've got a number of values, but, and I'm not probably supposed to pick one, but if I had to, it'd be pioneering, right? We have been the company that was first in the protein degradation space. We've got a long string of firsts, first to make them orally bioavailable, which means you can take it by a pill and not have it injected and things like that. We've been the first to get PROTACs to, cross the blood-brain barrier into the brain, first into clinical trials, first pivotal trials, first approval. Like, we have a long history of being pioneers, and that's the spirit, that's the culture, and that's what we have to now aim at in the future, and making sure that people have the next big horizon that we're aiming for is really important. And, as I've spoken to other CEOs that I've also taken on the role, in the past few years, what I hear over and over again is this is a role that's about people. It's about people management and inspiration and ensuring people can do their best work. So that's the stuff that keeps me up, is making sure that we're thinking of the things now that we have to think of to be in a different place in 2 years. So it's that sort of, what are we not thinking of? What are we not doing now that we should be? What could we be doing now that we're not? That's what I spend a lot of time thinking about.
Let's talk a little bit about the science at Arvinas. And let's start with PROTAC. Can you explain what this actually does? What makes this approach really different compared to a traditional small molecule drug?
Absolutely. A traditional small molecule drug, typically, and I'm going to generalize here, is a small chemical that essentially binds to a protein and prevents it from doing something. That's probably the simplest way to explain it. So you have one copy of the drug binding to one copy of the protein and preventing it from doing something, whatever that something is. It could be cutting something. It could be adding a phosphoryl group to something, a phosphate to something. It could be anything like that, right? The way that a PROTAC is different, which is our technology, a protein proteolysis targeting chimera, is that instead of just binding to that protein of interest, we bind to two things. One is the protein of interest that we want to get rid of, right? And then the other end of the PROTAC binds to part of the cell's natural machinery that's present in nearly every cell that degrades proteins. So essentially what a PROTAC does is it tells the cell, hey, we want you to degrade this protein. We want you to degrade all of it right now. Regardless of what you were going to do, that's what you want to do. So we are aiming at disease-causing proteins that typically are doing something that we don't want, or that there's too much, or that there's a mutation, or that they have a gain of function where they're doing more, they're something different than they're supposed to be doing. And there's a couple things about that that's really interesting. One is we're not just inhibiting the protein, we're getting rid of all of it. And that's important because proteins are not simple, and a lot of them have multiple functions. They might, in addition to cutting something, which is one example I gave, they might also help form a scaffold that other proteins can build around. If you inhibit just one function, you don't get rid of the other functions. So a PROTAC can get rid of all those. The other thing that I think is most important is, as I said, a traditional drug is essentially a 1-to-1 sort of ratio. You need a copy of the drug for every copy of the protein. A PROTAC is different in that when it binds to the protein and induces its degradation, the PROTAC can then be reused. It doesn't get used up or burned up or destroyed in the process and can go back around and do it again and again and again. So theoretically, we should be able to use less of a PROTAC drug to achieve a similar effect, which allows for greater potency and so on.
When you're degrading a protein like that, rather than just blocking it, what sort of diseases are going to become targetable that you couldn't target before?
Yeah, so it's the examples that I just mentioned where the disease-causing protein of interest is doing something unusual, or doing more than one thing. So as a couple examples, right? A lot of times you'll have a protein that causes disease, and then you get resistance to a therapy because the protein mutates. Great examples of this could be our first couple of programs. In androgen receptor for prostate cancer or estrogen receptor for breast cancer, there can be mutations in a protein where you want to degrade that mutant form. There might be other situations where you want to selectively degrade the mutant form, but not the wild type form. So those are great. Situations based on that catalytic mechanism that I mentioned, where each PROTAC can act again and again and again, that suits itself to a situation where you have a lot of a protein, or maybe where a tumor decides that in order to become resistant to a therapy, it's going to get turned on and amplified and get more and more of the protein made. That's a pretty common resistance mechanism. And because of the catalytic activity, a PROTAC can then churn through all of that extra protein that's made in a way that an inhibitor can't do. So for all of our programs, right from the beginning, we're asking the question, why do you need a PROTAC here? Because this is certainly not the case where a PROTAC, now there being an approved one, is going to supersede all other types of therapies. There will still be inhibitors, there will still be antibodies and ASOs and all sorts of things. They will all have their place. So the question is, why is the PROTAC the right tool for this job? And why is another tool not the right job? And that's how we think about all the programs when they start.
You mentioned that approval there. It's Vepdegestrant. That recently became the first PROTAC to get this FDA approval. What does that milestone mean for the company first, but also for this whole field of protein degradation?
Look, it's a huge deal, right? To put it simply, right? It's pretty unusual, and we celebrate like crazy in biotech when you get a drug approved. That's something that every person in biotech, you won't do that many times. The people that are the best at it will have a handful, single digits of approvals that they've been part of, right? And even rarer than that is to be the company that invents an approach, takes it all the way through discovery, through development to approval in a pretty short amount of time. But that's less relevant than the fact that, there's one company out there that is the first to get a PROTAC approved, and that's Arvinas forever, right? Just like there was a first company to get an antibody approved or anything else. It's a pretty short list. So it's an achievement that as a company, we're obviously very proud of. As maybe I'll call it the sub-industry of protein degradation companies, we're pretty excited about it because it really shows that a protein degrader can be a drug for sure. And I think as an industry, we're excited about it because of what it means for patients, right? It's a whole new methodology for attacking disease-causing proteins, which will lead, this will not be the last approval by any stretch, which will lead to new medicines being created for patients where other modalities were not able to achieve the right, mix of benefit and risk for patients. So it's ultimately huge for us, of course, but it's huge for us because of what it means for our ability as a broad industry to bring new types of drugs to patients, which there's unmet need all over the place that we can fill.
I'm glad you mentioned the patients there, because it's nice to hear the patients being brought up. It's not just an industry story. It's not just a great news for the company, but for patients too. You're developing that in partnership, or at least part of it in partnership with Pfizer for breast cancer. What does this mean for people who are already looking for treatments for breast cancer? Is this filling a very particular need there?
Yeah, absolutely. In the breast cancer space, the way it's evolved over the decades is that, patients go through different stages of treatment. And the stage of treatment that vepdegestrant has been approved for is second line metastatic with ESR1 mutations, following a CDK4/6 inhibitor, right? So a CDK4/6 inhibitor is the standard of care in first line metastatic breast cancer, where first line metastatic breast cancer is the first reoccurrence of the cancer after it's been initially treated. Typically, patients are treated with surgery and then go a long time on other drugs before it recurs and becomes metastatic. So our drug is approved in the second line setting. And the standard of care there for a long time has been a drug called fulvestrant, which does degrade ER, the estrogen receptor, but it does so with a very different mechanism. It doesn't directly recruit the body's natural cell degradation machinery. It's a bit more of an indirect method. And it's an injectable monthly therapy. And for a long time, the entire industry has looked for a better ER targeting therapy, which is what we've all been aiming for. And I think Vepdegestrant is a good example of. And so what it really means is that patients, when they reach that setting, they've got another option with that's been approved by the FDA, with an acceptable balance, with an entirely different mechanism than what else is on the market, what else is going to be available to patients. And in conversations with physicians, with patients, that optionality is really important. It's important to have options that work well, that work differently, and we're very pleased that that vepdegestrant will be one of those options.
One of the other options there, it's for a different indication, but ARV-102. That's the oral brain penetrant and PROTAC, which is targeting something that's implicated in Parkinson's disease and progressive supranuclear palsy. Getting a drug into the brain is already pretty hard. We've written about it this week at Labiotech. There are some companies trying different things. How does your platform actually tackle crossing that blood-brain barrier?
We get asked this question a lot, as the blood-brain barrier, as you said, has been a challenge to get therapies into the brain. And so we always get asked if we're using some sort of active transporter or some other mechanism of doing it. The answer is no. The answer is medicinal chemistry. The answer is that, just as our scientists were able to, back in the 2016 timeframe, able to figure out how to get a PROTAC degrader, which while it is a small molecule drug, they're typically a little bit on the larger side, which makes it more difficult to do things like get it into the brain or even cross the gut and be orally bioavailable so that you can take tablets by mouth. It's very similar to how our scientists figured out a way to cross the gut. They figured out a way to cross the blood-brain barrier too. So I can't go into the details of exactly sort of the rules of medicinal chemistry design that we use, but it's one that we've been able to solve. And we have shown preclinically and clinically now for ARV-102 that the drug gets into the brain, gets into the CNS, which is important because in the diseases you mentioned, which are, of course, diseases of neurodegeneration, we needed to degrade the target in the brain in the CNS. So that's a program that is in phase 1 now and just had some great data readout a few months ago. And then we'll be moving into later trials before too long.
And we mentioned Vepdegestrant, which you're working on with Pfizer. I mean, you've built partnerships with these big players. How do you think about that balance, that tension between keeping total control of the assets that you've got and actually bringing the resources of a big partner to bear to move towards commercialization?
You mentioned the right word right there, which is balance, right? Because our ultimate goal is absolutely to become a fully integrated company that's commercializing its own therapies. As a platform company, we've had great success moving great programs into the clinic. And that leaves us with a wonderful problem to have, which is how do you move them all forward? Even assuming success, how do you move them all forward? And partnerships have been an important piece of Arvinas work going back a long time. You mentioned the Pfizer partnership, of course, which has been critical for us since 2021. But we were doing target deals on the platform back well before I arrived in 2018. We had deals with Bayer and Genentech and Merck and Pfizer as well, and on that side too. And for me, the question is, what should Arvinas be investing in? I think we can do anything. I think we can't do everything, right? And so with multiple programs, I firmly believe that we will have to compete everywhere. There are no interesting non-competitive spaces. So if we're going to play to win, to take a strong role in any space, we're going to have to invest in multiple trials, multiple indications often, things like that. And making that commitment, going back to the topic of capital allocation before, means that other areas will get less. Well, I don't want other areas to get less. So if other areas are going to get less and the value of those programs can't be maximized by Arvinas, or we're not the right ones, then that's where a partner can step in. Using Pfizer as the example, success in breast cancer over the time when we did that deal in 2021 was not just second line, but also first line, also adjuvant therapy. And I'm not saying that Arvinas couldn't have done that, but if we had chosen to do that, we would have made different choices and we wouldn't be looking at the pipeline that we have right now. And if we're having this conversation again in a couple of years, I have no doubt that if you look at the pipeline we have now, some of those programs will still be, us going full bore at, and others will likely have partners to help push them along. So it's really about where can we broaden our ability to impact patients, or how can we accelerate it in a way that makes sense for a bigger company to do rather than us, which lets us then prioritize the big opportunities for us.
Yeah, I want to dive into that a little bit more because you've mentioned a couple of times this notion of capital allocation. Capital is to a certain extent limited at any point in time, and you've got to decide where to invest it, which program, which indication. How are you actually thinking about those choices? What are the sorts of priorities that you have in mind? Because when you've got a platform, it can go in so many different ways. That's a great advantage of the platform. But making that decision on where you want to invest your time and your money, that's really critical. What sort of things are you weighing up?
Yeah, and I think the word platform there is important, right? So I think Arvinas is probably one of the strongest examples of a platform company that you'll find, from the ability to take a technology and rapidly implement it into multiple programs. But make no mistake, as for a company of our stage, investors will look at us for the programs that we have in the pipeline. Program is wonderful, but I don't think anybody is going out right now and saying, investors, you should invest in us because we're the best at making antibodies or ASOs or some other modality. And so I don't expect anybody to look at Arvinas and say, okay, they make great PROTAC degraders, I'm investing in that. People right now are going to invest in programs. So that's a really important view for a small biotech, a small pre-revenue biotech, where we still rely on investors to fund what we're doing. So that actually helps narrow the field, right? Because that means that what we're taking forward and what we're known for has to be something that investors can get excited about. In my history at Arvinas, I've led both business development and investor relations. You can talk about the same program with big pharma companies and investors, and one group gets more excited than the other. This happens all the time. Like I will tell you for sure that the big strategic partners typically have a much longer term horizon for what they're interested in hearing about than investors do. And that becomes important because it means that what we're taking forward has to be investable in a way that is not necessarily the case for something that we're going to partner with a big pharma company. And the other thing to think about is, it'd be really nice to, take everything to late phase 2 or phase 3 and then partner it. That gets pretty expensive pretty fast. So the other thing that we try to do is identify what data do we need to generate to get ourselves excited and make decisions, to get investors excited to help with financing, and to get partners excited. Because whenever a company announces a partnership, it's not like it just came up a month ago. Those conversations, I'm sure everybody would tell you, are the result of years of work, right? I'll give you an example. We kept many big pharma companies up to speed on our progress with our androgen receptor degraders for a lot of years before we ultimately partnered with Novartis in 2024. That didn't just happen. Those conversations had started before I joined the company in 2018. And so as we think about prioritizing what we do versus partners, all that comes into play as sort of what's the triangulation between what we can invest in, what a partner wants to invest in, what investors want to invest in, and that's the complicated math that we're doing.
Let's look forward a little while because it's a long-term play. Pharma, just by its nature, is a long-term play. If the platform delivers on the sort of promise that it already has started to deliver on now, what is it actually going to change for patients? What's the version of the future that you're working towards?
I think PROTACs, as I said before, we try to think about where a PROTAC is the right answer. And we don't presume that the PROTAC is the right answer for every disease that's out there. If we're doing it right, patients will have therapies for disease targets that they don't have therapies for. There will be, first in class or therapies where there's nothing else that works. I think the most obvious examples of those right now are our work in neuroscience, in neurology, both for the LRRK2 degrader, which you mentioned is relevant in PSP and Parkinson's. There are no disease-modifying therapies in PSP and Parkinson's, either one. So if we could actually create an oral small molecule that changed the course of disease, wow, that is a dramatically different world for a patient diagnosed with one of those diseases. Another example is our work in Kennedy's disease. We just started that program this year, in the clinic rather, just started in the clinic this year. That's a disease that takes decades to progress. Men with Kennedy's disease have no options right now that will slow the progression of disease. And a PROTAC there could do something that other drugs just won't be able to do. So I think that's it's of course good when the industry moves in steps and things get better over time. Like, it isn't that common that the paradigm gets radically changed with one therapy. Usually it's a stepward progress over years, and that's good. But I think PROTACs will also lend themselves to big leaps where something that wasn't possible before because an inhibitor didn't hit all the functions of a specific protein or wasn't specific enough, where a PROTAC can do that. And I think that ultimately that'll be measured by drugs for diseases that nothing else works for.
I guess if that's looking forward for, 10 years' time, what about a little bit shorter term, the next 12 to 18 months, and if we're talking about Arvinas, what sort of milestone should we be looking for? You mentioned a couple of trials that are meant to read out this year.
Yeah, it is a really intense period of time for us. I would say starting a few months ago, going to the end of next year. A few months ago, we had that phase 1 data for the LRRK2 degrader, which is relevant for PSP and Parkinson's. That's one where we've gotten the data from that phase 1 trial, and we're aiming at starting the next wave of trials for that drug by the end of the year. When it comes to data, all the programs that I've referred to all have data coming in relatively short order, including some programs that I haven't mentioned yet, right? So I mentioned the SBMA program, or SBMA is another word for Kennedy's disease. That'll have its first phase 1 data in the first half of next year. We've got a degrader that I haven't mentioned yet for an NHL target, non-Hodgkin's lymphoma, called BCL6, which will have its first phase 1 data later this year. We are starting a trial in the second half of this year for a protein called a degrader for HPK1, which is our first immuno-oncology target. We haven't guided towards data for that yet, but it's a trial that's going to start in the third quarter this year. So you can imagine by the back half of next year, we should be seeing early data from that too. So it's a period where all the work that we've done to refill the pipeline, sort of after our initial success with androgen receptor and estrogen receptor, that all came into the clinic in the past couple of years, and it's all going to read out in its first phase 1 trials, which will then enable all the work that we just talked about around how do you prioritize, what does Arvinas do, what do we take forward, what do we partner for, how do you raise, all those questions come up very rapidly in that time window of 12 to 18 months.
And so for anyone who wants to follow along, anyone who wants to keep track of these milestones, where can they go to keep up with you? Where can they learn about the science? Where can they keep up with the business?
Well, we try to be pretty proactive about getting our word out. The website's probably the first place I would go, but we're pretty steady on social media with making sure people see press releases that they might not get in their news feed. We have spent a lot of times, if you're on the science side, we are very careful to get out and talk about our data at scientific congresses. If you're on the investor side, we take a lot of care to make sure we're at investor conferences talking about what we're doing. And if you're anybody else, I think this podcast being a great example, we're trying to make sure that people across media spectrum know what we're up to and can see the progress that we're making. It's important to us that we set out, tough to achieve goals and then make sure that we celebrate when we hit them. So I think a lot of ways to keep up with the progress that we hope to make in the coming years.
Randy, it really sounds exciting. Thanks for joining us today on Beyond Biotech.
Thank you very much, Dylan.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.